Nessa palestra, I will be talking about different career paths in veterinary microbiology. Then I will present some research examples of what we can do with the free data generated daily in diagnostic labs, and also how can we apply DNA sequencing in a microbiology diagnostic lab. So to start with the first topic, different career paths in veterinary microbiology. To start this presentation to talk about my career path, I think it's very important to share with you my desire to find a career path that was meaningful to me. I grew up in a rural area in the south of Brazil, in Rio Grande do Sul. The name of my small town is Constantina. And as I was deciding what to study, it was, of course, veterinary medicine, agronomy were like in my top choices because of my background and what I found that was really meaningful to me. I had the genuine desire to help local farmers in my region. So another reason that I think was really important to me when I decided to study vet med and why I am still working on veterinary microbiology and what I do is not only because I have my sister who is also a veterinary microbiologist, is also because I heard the story of Alexander Fleming, who was a Scottish clinical microbiologist. He was working in London at St. Mary's Hospital, and there in his tiny lab, he discovered penicillin by accident. I was really fascinated by his power of observation. So then, since my first semester in vet school in 2007, I looked for an internship to work in the mycology lab because I was really fascinated about penicillium and Alexander Fleming. And that's how I started this journey in microbiology. Later, I worked in the bacteriology lab. And because of this internship, what was really the turning point in my decisions and my experience in diagnostic microbiology was because in my last semester, I got a scholarship from CAPES to spend 6 months at the Athens Veterinary Diagnostic Lab in the University of Georgia in the US. And during my vet school, I guess I'm talking to several students that are aspirants of microbiology or infectious disease from Rio or from other parts of Brazil. And I know that sometimes we don't really see the light at the end of the tunnel. And I really thought that there was no market for me in Brazil or in other places. I did not know what I could do with the experiences that I was getting in the microbiology labs from the Federal University of Santa Maria. But when I went to Georgia for these 6 months, I could realize the potential and all the doors that could be opened to me. So then I did my master's also in Santa Maria. I was investigating the antimicrobial resistance of different species of Moraxella that causes bovine keratoconjunctivitis. And then I moved after my master's to England to take my PhD at the University of Nottingham, where I was studying ovine foot rot, investigating specifically the microbiome and the innate immune response against this sheep disease that is very, very prevalent in the UK. So at the end of my PhD, as I was doing like a full research PhD, I was not involved in diagnostics. I was really missing the microbiology diagnostic world, and I was trying to find ways that I could go back to that environment. Not that I don't like research, I love research, but I was really like trying to find a way that I could do research applied on diagnostics. And as you know, veterinary microbiology, or for those that are not in the field, is a quite narrow field, and we tend to know each other. So I knew that in the US, there were 1 or 2 residency programs in microbiology. I knew that in Brazil, some universities were starting to offer residency programs in preventive vet med or infectious diseases, but things were not very established. In the UK, things were starting as well, so I just decided to contact my previous supervisor, the professor that received me when I was a veterinary student in 2011, and I said to her, I want to work as a resident in microbiology. I know there are not many programs in microbiology in the world, and my dream is to take the board certification exams from the American College of Veterinary Microbiologists. And I was inspired by people that were close to me that were veterinary pathologists. And I could see that in the area of veterinary pathology, things were really well established in the US and in other parts of the world. They have residency programs, they have board certification exams that are very well established. And then I was wondering why I don't try to do the same for veterinary microbiology. So the professor from Georgia, Dr. Susan Sanchez, she said to me, yes, we don't have a residency program, but what if we create one? So that's exactly what happened. They created a residency program. I was the first resident in Georgia, and I believe one of the first residents in the USA to take like a full-time residency in microbiology. And for the students that are here listening to me and wonder what does a resident in microbiology do? So what I tried to do with my supervisors from Georgia was to mimic the residency program from pathology programs that are very well established programs in vet med. And I was doing like lots of different activities, including like preparation for the board certification exam. This is a very special, I would say, very special process that certifies different specialists within different areas of vet med. If you have questions, if you would like to hear more about the American College of Veterinary Microbiologists, please email me. I'm very happy to talk about the process and how I managed to sit for these exams. And so I was studying, preparing for the exams. And also, I think one of the things that was a key point, and I think that is one of the main roles, jobs of a veterinary clinical microbiologist, is client communication. So, for instance, I was in a diagnostic lab that was processing lots of samples and sending results to clients. These clients are usually veterinarians, large or small animal clinicians. And they did not know how to interpret many of the reports because sometimes we used to identify very unique species of bacteria. And so that is, I feel that is one of the main roles of the clinical microbiologist, is to help other veterinarians to put the pieces of the puzzle together, to put all the information of a clinical case and help them to reach like a definitive, reliable diagnosis. So that is one of the things that I was doing, talking to our clients every day. Also, I was helping with the management of the diagnostic lab, and of course, processing samples and analyzing samples from the bacteriology, mycology, and virology labs, as well as the molecular biology lab. So if you are interested, just let me know. I can share more information with you. So I just finished the residency last year in 2020, and now I am an assistant professor at Ontario Veterinary College in Canada. And here we have like 2 different images of what you can expect from Guelph in Canada if you ever come here to visit us. I'm here for less than a year, but I have seen like both pictures. So we have like very 2 completely like different scenarios during winter and during summer or fall. Okay, so the next part of this presentation is something that is very special to me, because I feel that as a Brazilian, I was trained under lots of different limitations, I would say, in terms of how much budget, how much money was available, and the resources that was available in Santa Maria to do different types of research. So I understand the budget limitations, and I always try to do, since the time that I was in Santa Maria, I was trying to pull the data from the diagnostic workup, and try to see if we could somehow build a story, a nice story, and publish that story. So that would be like a free data, and we could have like a nice research project, and we would not need to spend too much time on research. So in Georgia, I would like to share 3 stories, and perhaps these may inspire residents from the microbiology residency program, or graduate students, or even like veterinary students or clinicians that work in the veterinary hospital. Perhaps you can be inspired by these stories and the way that we analyze the data and we publish this data. So the first story that I would like to share with you, it started with a client question. And the question from this client, there was a veterinarian, of course, was, what does PCR test positive for Mycoplasma SPP mean? And that was not only from one client. I heard this question from several clients, because the PCR test that we are offering in Georgia for Mycoplasmas was not speciating, like was not identifying the species of Mycoplasma. It was only Mycoplasma spp. There are thousands of different species of Mycoplasmas, so perhaps they were all positive because we were detecting a commensal Mycoplasma. So I was figuring out that there was an issue with the PCR testing that we were offering, and I was searching the literature, and in the literature was not very clear the different species of Mycoplasmas that are associated with canine respiratory diseases, for instance, neither for feline respiratory diseases. But this story is specifically about canine respiratory diseases. So that's how it started. And then I started to read more about kennel cough, that is in Portuguese, I think is called tosse dos canis, pelo menos, at least in Rio Grande do Sul, that's how we call. So this disease, it was quite prevalent in Georgia. I was very surprised by the number of positive PCR for the different pathogens associated with kennel cough. And the disease is caused by different bacteria, different viruses. It's a very complex disease. And there were a few questions that we did not know at that time. And then I thought, perhaps we can pull the diagnostic data together and we can investigate, we can try to answer these questions that are like unanswered questions in the literature. So these questions were, what we did not know before starting this project: which co-infections I mean when we have the presence of a bacteria and a virus together, or different bacteria and viruses together causing disease, which of these combinations of these co-infections are more common? And which of these co-infections, of these combinations, would lead to a more severe clinical presentation, to more severe disease? For instance, would lead from coughing to a pneumonia. And of course, all this started because of the role of different Mycoplasma species in the canine infectious respiratory disease. So what I did was something very simple. I think anyone can do that. Here is the list of pathogens that were included in the respiratory PCR panel. It means that every time that a client used to submit a swab, for instance, a nasal swab from a dog for diagnostics, we would offer a PCR panel. The cost is around $100 American dollars, and the client would receive the PCR results for all these pathogens that are potentially associated with this disease. So I just went to the database online and I retrieved for the past 10 years all PCR testing that we did for canine respiratory disease that ordered this PCR panel. And then I found great collaborators that are statisticians, and they helped me with the statistical modeling analysis. So to answer those questions, the results that we found were that Mycoplasma sinus, Mycoplasma canis, these two species of Mycoplasma, canine parainfluenza virus and Bordetella bronchoseptica, they were the pathogens that were more commonly detected together, causing co-infections, which was very different from canine distemper, from coronavirus, from adenovirus, and from influenza virus. So another question was, which of these combinations is the worst combination? For instance, if a veterinarian receives a report from a diagnostic lab with these combinations, what is the worst prognosis for a dog? The worst combination is canine parainfluenza virus with Mycoplasma sinus. So if we have these two pathogens associated with this disease, causing disease in a dog, this dog tends to develop pneumonia. And we believe that this information may help clinicians with prognosis of this disease. We also found that young age is the most important predictor of disease severity. And as I mentioned, Mycoplasma sinus and Mycoplasma canis, these two species of mycoplasmas are more associated with disease. So what we did, the practical application of this study was to develop real-time PCR multiplex that detects Mycoplasma sinus and Mycoplasma canis at the same time. And this PCR was added in the PCR panel and is now being offered to clients. So here is the publication, and I have to acknowledge Dr. Mauricio Seguel and Dr. Roy. They were like helping me with the statistical modeling analysis. Without them, we would not have this data so well analyzed the way they did. Okay, so the next story that I would like to share with you is about Acinetobacter and how this story started. It also started with clients' questions. They were calling the diagnostic lab and asking, what does positive culture for Acinetobacter SPP mean? And sometimes using MALDI-TOF, we could identify the species, specific species of Acinetobacter. And that was even worse. So then they were like totally lost. Probably in vet school, they have never heard about Acinetobacter. And the literature in veterinary medicine about Acinetobacter is quite scarce. There are not many papers out there. So there were quite a few clients calling us to ask, what does it mean? Do I have to use antibiotics or not? Does it imply that it is causing disease or not? What is the role? So I started to pay more attention in Acinetobacter. And so just like to give you like some highlights, Acinetobacter is very well known in the human aspect, and we don't know much in vet med. As I said, according to CDC, it causes around 500 deaths per year in the United States. We don't know exactly the role of different species, and the question was, is it an emerging pathogen in veterinary medicine? Is it as emerging as in human medicine, as we can see this data here from CDC, from the human side? So what did you do? We retrieved data again from the online database from the diagnostic lab. We asked the right questions that were like open gaps in the literature, and we applied the right statistical modeling. So to answer some of the questions that we asked, we found that Acinetobacter, it is a significant microorganism, and we also found alarming multi-drug resistant rates. We know that in humans it is quite alarming, but we didn't know much about animals. And most of the isolates, they were from domestic pets, highlighting the presence of antimicrobial resistant Acinetobacter in the human environment. We don't know if Acinetobacter is a zoonotic pathogen, how this transmission occurs, if it occurs, but I think it is important to highlight that it was mostly like from domestic pets, actually mostly from dogs. So we could see that Acinetobacter baumannii, that is the most prevalent species that we found, was found was highly prevalent in dogs, and these dogs were more likely to present Acinetobacter with multi-drug resistant phenotypes. So this study was just published last year, and for microbiologists that are watching this presentation, they may wonder what is like a more practical contribution of this study. So in this study, we suggested a few points to CLSI. What is CLSI? Is the Clinical Laboratories Standard Institute. So in the diagnostic microbiology world, I would say that CLSI is like the Bible of microbiology. So most of the tests that we run, the interpretation to say if a bacteria is resistant or susceptible is based on these guidelines. But these guidelines don't have anything about Acinetobacter from animals. They only say, and they only give suggestions and recommendations to Acinetobacter from humans. So that's why it is important to investigate Acinetobacter from animal origin. So we highlighted in this paper that there is a need to determine specific breakpoints for antimicrobial susceptibility of Acinetobacter from animal origin. So veterinary labs, they can use like a more reliable information to say if this bacteria is susceptible or resistant. We also highlighted that the intrinsic antimicrobial resistance from human Acinetobacter baumannii isolates is totally different from animal isolates. So we have to interpret their guidelines like very, very carefully. So these are some of the points. Okay, the next story that I would like to share with you is about how we can use sequencing technologies for the identification of novel pathogens based in a diagnostic context. And this story, unfortunately, is quite sad. So how all that started? So this story started with a cat. His name was Bubu. And when he was alive, the veterinarian submitted a biopsy for histopathological examination. The pathologist provided a wrong diagnosis of Cryptococcus. So the owners were very concerned about the potential zoonotic, if Cryptococcus was potentially zoonotic or not. So the owners decided to opted for euthanasia of this cat based in this on this wrong report from the pathologist. So after euthanasia, the cat was submitted to the University of Georgia for postmortem, and that's how I entered in this story. We received a piece of the nasal tissue from this cat. We can see that the nasal planum was disfigured, and the request I remember very well was, we want to speciate Cryptococcus, we want to define the species because of potential zoonotic concerns. And I was like, okay, straight, let's investigate Cryptococcus. Perhaps they already know that it's Cryptococcus. So what we found on the lactophenol blue stain was this structure. This is certainly not Cryptococcus. We can see here a sort of sporangia, and it reminds Prototheca, but we are not sure about if it was like Prototheca or Prototheca or not. So in the lab, we have Sanger sequencing that can be applied for rare cases like that, different cases that we don't reach diagnosis, like based on morphological identification. And the Sanger sequencing, that is a sequencing technology that is going to identify, to sequence a small fragment of a specific gene, came out as if this organism is actually Prototheca curis. So I went to the literature, I was like, Prototheca curis, what is that? There was no report in veterinary medicine at all. And then I was thinking that perhaps we did something wrong. So we repeated, it was again Prototheca curis. The only publication that I found was from a man from Japan that had like subcutaneous skin lesions. And from there, they identified this new species of Prototheca called Prototheca curis. So I was like, perhaps that is the first report or the first time that we are identified that in VetMed. So we did also whole genome sequencing, and we did a comparative genomic analysis. We confirmed there was really Prototheca curis. And then this story was just published in Emerging Infectious Diseases. And that is the first report of Prototheca curis infection in VetMed, as I mentioned. I brought this case and this example I wanted to share with you because I think here the take-home message is the importance of communication, not only with clients, but communication between pathologists, microbiologists, and small animals or large animals veterinarians. So I feel that sometimes we tend to work like isolated, but I think this is an example of how important it is like to combine different expertises to reach like quite rare diagnoses like this. So that is the take-home message, that we should talk more, we should work better together. Okay, so the final part of this presentation is about DNA sequencing technologies. And all this started, of course, with another story. As I was doing my residency in Georgia, they asked me to help with the initial validation and optimization of sequencing technologies in the diagnostic lab. It was like the years that sequencing was like all veterinary diagnostic labs in the US, at least, were starting to implement Illumina sequencing, Sanger sequencing in their routine. And as I was doing that, I received a call from a client, another client. And a research project started because of this call. And it is associated with super multi-drug resistant, fastidious, slow-growing bacteria. So here we have an example of how this clinical case looked like. It was a cat with several, many draining tracts. The wounds were very severe, and the veterinarian had never seen something like this, and the vet that was our client was calling us quite a lot because he was quite concerned about what he was seeing, treating with different antibiotics, and that was not working. And then he submitted a sample. We did PCR for all possible bacteria or fungi, pan bacteria and pan fungal PCR, and we had a surprise. What we got there was Mycobacterium abscessus, which is from the same genus of Mycobacterium tuberculosis or Mycobacterium bovis that causes bovine tuberculosis, for instance, but is a different species. And this Mycobacterium is also slightly different because it grows a little bit faster than other Mycobacterium, for instance, Mycobacterium bovis or tuberculosis. So here, just to remind, Mycobacterium is Azil-Nielsen positive or acid-fast positive bacteria. And the veterinarian was like, really, I need to know which antibiotic would work or not for this case, because I have tried two different antibiotics that were like marbofloxacin and clarithromycin, and the animal was not presenting any improvement. So the issue here is that susceptibility tests for certain bacteria, for instance, for Mycobacterium, may take a lot of time, sometimes almost a month, according to the Bible of a microbiology that I call CLSI. So meanwhile, the client was waiting for the results. Almost a month later, we submit the results, and here we have a surprise. Clarithromycin and marbofloxacin were resistant. So I started to wonder, is that a faster and still reliable test to predict antimicrobial resistance that is not based on the standard traditional way of identifying antimicrobial resistance? Probably you are aware of disk diffusion or broth microdilution methods. These are the standard traditional methods. And I was wondering, perhaps PCR, if we detect a specific resistance genes, but if we use PCR, that is going to be just how we're going to know exactly which gene to target to have like a PCR for each different gene. As I mentioned, at the same time, I was working the implementation of sequencing technologies in the lab. And then I was like, okay, let's try perhaps to use sequencing to provide a list of possible antimicrobial resistance genes. And that would somehow help in cases of fastidious, slow-growing bacteria. So for those that don't work on sequencing or are not aware on sequencing, I just brought this slide to highlight what is the names that I am using during the presentation. So we have Sanger sequencing that is considered first generation. This was the first one. It used to take a lot of time. It was very, very expensive. Now it's extremely cheap. With around $10, we can run Sanger sequencing. So most of the diagnostic labs in North America are implementing Sanger sequencing and offering this service to clients. Then we had a second generation sequencing. Now we are in the third or fourth generation sequencing. Some people say we are in the post-sequencing era. And during the presentation, I will be talking about these technologies. The gold standard that we have nowadays is Illumina sequencing. We also have PacBio, and we have Nanopore. That is a new technology that was introduced, I think, 2014, 2015. And when I started to hear about nanopore, I was like, wow, that is really fascinating. I think that is going to be the future of microbiology. Let's wait a few years to see if that is going to happen. The idea is really, really fascinating, and that fits really well the demands of a diagnostic lab. So, as I mentioned, I was looking for other alternatives, and the idea of nanopore technologies was fitting really well with that idea that I had, where else we can use. So here we have nanopore. So nanopore is this technology that has several advantages. For instance, we can prepare the library prep quite fast. It's quite easy to manipulate the data that is generated. And what is more fascinating is that it's really, really small. That is the machine. It's really, really small, very light, and it's portable. So we can use this machine in Antarctica. We can use in the field. We can use in the lab. We can use anywhere. So it's really, really fascinating. I think for diagnostics, the idea fits quite well. And what is really interesting, at least to me, is that we can identify in real time as we put the sample in the sequencer, we have a real-time identification of the bacteria or the virus or the fungi that is present in the sample. And we also have the identification of antimicrobial resistance genes. Of course, there are lots of challenges associated with that. Nowadays, we face challenges. For instance, the price is still quite expensive, mostly like for veterinary diagnostics. And another challenge that I was facing when I started to work with that, I was like, okay, if I want to use this to identify antimicrobial resistance genes in the whole genome of the bacteria, which DNA extraction do I use? And my question is because here we have the bacterial cell, and we have the chromosomal DNA and the plasmid DNA. And we know that most antimicrobial resistance genes, they are located in plasmids, not only in the chromosome. So I was like, if we use just a normal, I would say, QIAGEN DNA extraction kit that are quite popular, can I extract all plasmids that are present in the bacterial cell so I will have a complete list of antimicrobial resistance genes? Because if I miss some of these plasmids, I may miss important genes, therefore wrong information to potential clients. So I was wondering, we need to know exactly which way we have to extract the DNA from these samples. Another challenge is that using genomic technologies for antimicrobial resistance means only prediction. We cannot say that the gene is going to express the phenotype, for instance. So if we detect the gene, does it mean that we are going to have the phenotypic antimicrobial resistance that is also detected, that is commonly detected by standard methods, for instance, disk diffusion? So that was another question. Does it correlate with phenotypic data? So just like to highlight some of the aims of this study, we were developing like a sequencing workflow for reliable MR prediction. And at the same time, we were like investigating the ideal sample preparation to identify antimicrobial resistance genes. Do we need a plasmid extraction step or not? And does it correlate with phenotypic tests? We did all that with a wonderful microbiologist specialist in plasmids, Dr. Ann Summers from UGA. So these results, if they correlate somehow, is because she was behind the scenes helping me. Okay, so results from the first goal of this study, we wanted to optimize a fast and reliable workflow for detection of antimicrobial resistance. So for instance, day one, we receive a sample, let's say a swab from an animal. In the day two, if it is a fast growing bacteria, we have this bacterium in culture. Then if we are working in an automated diagnostic lab, we can use MALDI-TOF for identification of the species, for instance, and we can do antimicrobial susceptibility tests in an automated way as well. So we think like the second day we can have all these results. Then in the second day, if you want to work very hard in that day, we can do DNA extraction. And I was wondering if we have to also do a plasmid extraction. And then in the third day, we just prepare the sequencing, and it usually takes 20 minutes just to prepare the sample. It's really fast. And to sequence, for instance, a bacteria like E. coli, a small genome, we can do within 6 to 8 hours. And then after that, we may have a list of antimicrobial resistance genes that may guide clinicians in this third or fourth day. Here we did everything for fast-growing bacteria, but something interesting to do that we are aiming to do in the future is to optimize this for fastidious, slow-growing bacteria and anaerobic bacteria. In addition to do that, since Illumina is the gold standard technique, we wanted to compare all these that we got from nanopore with Illumina. So we could have like a very good comparison of the genes that we are seeing. And here I just delete the plasmid extraction step because I show you that we don't really need to waste 6 hours to extract plasmids for sequencing. And then the question was if plasmid was really necessary. It takes time. Is it necessary to combine nanopore and Illumina sequencing? Illumina, as I said, is the gold standard. It means if I add another sequencing technology, that is quite expensive. So to answer the question from the second aim of this project, we could see that, for instance, here for E. coli, all genes that we found in the plasmid extraction were also present in the total genomic extraction using like a normal QIAGEN DNA extraction kit. So we don't really need the plasmid extraction step. It means it saves 6 hours. So we can say that the plasmid extraction is not essential to obtain a complete list of antimicrobial resistance genes. The last aim was, do phenotypic tests correlate with genotypic tests? We could see that yes, all genes that we identified had a phenotypic resistance determinant. So resistance genes corresponded to all categories of phenotypic resistance. Okay, just a summary of this study. The workflow took around 3 days. I would say 3 to 4 days to have like a full analysis of everything, unless you want to work really hard during 3 days. The cost per sample, I would say that is around $120 per sample. It still costs this like budget is an issue. But with this, we can have like a resistance profile. Resistance profile, I mean all resistance genes present in a bacterial strain. Do we need to use a plasmid DNA extraction? No. The specific kit that we used for total DNA extraction could predict all genes that we could see in the plasmid. It means that this kit also extracted all the plasmids and the chromosomal DNA. Therefore, using only one kit is cheaper and faster. And we could see that all genes corresponded to all phenotypic categories. And do we really need Illumina, the gold standard technology? No. We found out that to answer this question, the main question here, only with nanopore, there was enough for the strains that we used. Okay, so going back to the point that this story started. And here is that a faster and still reliable test to predict antimicrobial resistance? If you remember the case of the Mycobacterium abscessus, we can say that nanopore technologies and other sequencing technologies are there. They are not ready yet to be applied in a diagnostic scenario. I know that many private diagnostic labs in North America and some human diagnostic labs, they are already using next generation sequencing for antimicrobial resistance prediction or for pathogen identification. Unfortunately, I think that in VetMed, it may take still like some time, but hopefully you get there. Okay, what are the take-home messages from this research seminar? I think the main message that I wanted to bring is that microbiology and molecular biology research does not necessarily need to be expensive. We know that molecular biology kits and reagents tend to be quite pricey, but perhaps if we can just reuse the data that we are producing in teaching hospitals, in veterinary clinics, or in diagnostic labs. If we can combine all this data, we can answer quite important questions that are open gaps in the literature. And also, I would like to highlight how precious the results that are generated in a daily basis in laboratories and veterinary clinics, they are. So let's make the most of them. And I think the key point is identify the gaps in the literature, ask the right questions, and apply the right statistical tests. And my take-home message to the students from the Federal Rural University of Rio de Janeiro is that find a career path that is meaningful to you. Do what you think is really important, what connects you with something that goes beyond and help the community that you are connected with. So I think that when you find what is really meaningful to us, things will just unfold naturally. And I hope you can find a career path that is truly meaningful to you. My acknowledges, and muito obrigada pela oportunidade de falar um pouquinho de microbiologia com vocês. Por favor, me contactem por e-mail. Ficarei muito feliz em poder me comunicar com vocês.